So hi guys, welcome to the Obscure Scholar YouTube channel. So before we begin, I want to make one thing clear. This video is primarily for my own consistency and active recall. I have oftentimes I've heard that when we are reading a book, we tend to remember only 10%, whereas if learning is both audio visual, for example, a teacher is teaching us, that is both audio visual, so then we tend to remember about 20%. But when we are actually teaching someone, so then only the maximum, the understanding and retention happens. So that is my main purpose, to maintain my own consistency and for my own revision. So yeah, let's start this video. So we'll start with cell biology. So what is a cell? Just a second. Okay, so what is a cell? Cell is the basic unit of life, right? So we might also call it as fundamental, uh, structural and uh, fundamental unit of life. So what are the points through which we can call something a cell? So a cell must have three basic criteria. That is, first of all, we it should have a cell membrane or plasma membrane. Then it should have, cytoplasm, and then it should have genetic material. That is DNA and RNA. Right. So it must have cell membrane, cytoplasm, and genetic material. So what is cytoplasm? Cytoplasm is a so yeah, cytoplasm is a fluid matrix where all the chemical reaction happens. Here, cyto means cell and plasm means fluid matrix. So then, if we do some word breakdowns, what will be the meaning of cytogenesis or cytolysis? So cyto means cell, as I said, and genesis means generation. So cyto generation will mean generation of a new cell, or when a new cell is created. And lysis means death. So cytolysis will mean death of a cell, right? So let's proceed further. Let me draw a eukaryotic cell. First, let me draw three structures. So this is a lysosome. Let's call this let's make the lysosome here. Here we have mitochondria, and here we have a vacuole. So what happens here is, in lysosome the breakdown or digestion of food happens. Okay, and these food is taken by mitochondria to generate energy. That is why mitochondria is, we all know, it is called the powerhouse of the cell. And the remaining food, or this vacuole acts as storage. Whatever is left gets will be stored in the vacuole. So lysosome will help in digestion, and those digested things will go to mitochondria in order to generate energy, and lysosome will act as a storage medium. So in mitochondria, what happens? Aerobic respiration happens. So how this generates energy? So if we see, so what is aerobic respiration? It is glucose with oxygen gives rise to energy. So what is glucose? C6H12O6 plus, we take oxygen, then with here we'll have some activation energy in the form of an enzyme which is made by ribosome. We'll come to that later. So what do we get? We get CO2 plus H2O plus energy. So yeah, so you can see, through aerobic respiration we are getting this energy. So now here we have a nucleus. Here all the genetic materials are present. Then we have ribosomes. By the way, ribosomes are non-membrane bound organelle. Here, if we see mitochondria is membrane bound. Lysosome is membrane bound, but ribosome does not have any membrane as such. So the main function of a ribosome is protein formation, and by protein here I mean enzyme. So all enzymes are basically proteins only, but all proteins are not enzymes, right? So then we have these structures which is known as endoplasmic reticulum. And when these ribosomes they get stuck to this lower portion of this endoplasmic reticulum, we call this as rough endoplasmic reticulum, and the smoother surface where ribosomes are not that much present here, this is a smooth endoplasmic reticulum. The function of endoplasmic reticulum is transportation. So the rough endoplasmic reticulum helps in transportation of proteins, whereas this smooth endoplasmic reticulum, it transports lipids and fats. So yeah, and over here we'll have another organelle known as chloroplast. And chloroplast where chlorophyll is present. Of course, this will be only there in a plant cell. So yeah, and we have another organelle. These small organelles will be there known as Golgi body or Golgi body, whatever you feel like calling it. Golgi body. So Golgi body helps in packaging. We might call it as their function is import export. Okay, so it just does the packaging, and the nutrients are sent outside or inside. So that is done through Golgi bodies. So yeah, now let's go to nucleus. Okay, so in nucleus, it is the brain of the cell, and here genetic material is present. So genetic material are DNA and RNA, right? So how does the nucleus controls the entire function of a cell? By controlling the enzyme which needs to be formed, right? So by controlling the enzyme, it controls the overall functioning of a cell. And what is the function of genetic material? It acts as a storage of information. Every information is stored inside the genetic material. For example, we'll discuss it further also, how a ribosome will make a certain form of protein. So that information, that particular enzyme needs to be made, that information will be given by the genetic material to ribosome, and then ribosome will make that particular enzyme, right? So yeah, let's proceed further. We'll start with genetic material now. Okay, so let's understand about genetic material. So in genetic material, there is DNA, there is RNA. So RNA, DNA is deoxyribonucleic acid, and this is simply ribonucleic acid. So why deoxy? Deoxy is there because the oxygen molecule has been removed, right? And ribose because this DNA contains a sugar, right? A sugar named ribose sugar. The structure is like this. Structure of ribose sugar, right? So yeah, and why nucleic? Nucleic because it has four nitrogenous base nitrogenous base compounds present. That is, there is adenine, we have guanine, we have cytosine, and we have thymine, right? And in RNA, this instead of this thymine, we have uracil, right? Everything else would remain the same. We'll have adenine, we'll have guanine, we'll have cytosine, but in place of thymine, we'll have this uracil. Why acid? Because DNA is generally acidic in nature, right? So let's see how DNA looks like. So it generally looks like this. It's a double spiral, doubled helix structure, and within here you'll find small lines. These are basically hydrogen bonds, which we'll see. So these are hydrogen bonds between the nitrogenous bases. So if we open the strands, what we'll find is something like this. I'll draw the diagram as well. So they are present in a series, like for example, it is there in a sugar phosphate manner, right? There will be one sugar, then will be one phosphate, then another strand will be there will be one sugar phosphate, and within that we'll have double bonds and triple bonds of these nitrogenous bases. I'll just draw it and show you. So it will be somewhat like this. If we This is sugar, then we'll have one phosphate, then again we'll have that ribose sugar, then CH, right? Then again we'll have a ribose sugar. So this is how the entire thing is present. The sugar phosphate. Above also we'll have structure like this. Here there will be a phosphate molecule, right? So it will be present like this, and here the nitrogenous base will be present. For example, if there is thymine, here if there is suppose adenine, right? So why I'm drawing this structure, I'll explain it. So here we have suppose cytosine, right? So in front of thymine we'll always have adenine, right? Adenine and thymine will always face each other, and cytosine and guanine will always face each other. So here if there is thymine, we'll definitely have adenine here, right? So now here the bond which is present, which binds these adenine and guanine, is a double bond. Okay, yeah, there will be a double bond, and here if we see there is adenine, so of course here we'll have a thymine. So again these two will be connected through double bond. Here we have cytosine, right? So cytosine will make a triple bond with what? With guanine. So we have a G over here, and we'll have a triple bond. So these are hydrogen bonds which are represented over here, right? So yeah, as I said, uh, adenine will always form a double bond with guanine. Sorry, adenine will always form a double bond with thymine. Thymine will always form a double bond with adenine, but cytosine will form the triple bond with guanine, and same as well vice versa. G will form a triple bond with cytosine, right? So why these I have made a square here, and here I have made this sort of a structure. So adenine and guanine are together known as purines. and cytosine plus thymine are together known as pyrimidines, right? So yeah, so now that was the basic structure of, just let me add a page. So that was the basic structure of DNA. Now we'll see. So the DNA strands are here. The nitrogenous bases will be randomly arranged, right? A, T, G, we'll have C, then maybe again we'll have C, then we'll have G, sorry, we'll have G, then T. So in front of adenine, what we'll have? Thymine. In front of A, what will, T, A, we'll have A. In front of G, we'll have C. Then again a G, again a G. Here, if it is G, we'll have C, and here is A. So here we'll have a double bond. Here we'll have a double bond. Here there will be a triple bond. Similarly here, triple bond, triple bond, triple bond, double bond. So why I am showing you these? Because now why DNA is known as the what should I call it, it stores all information, and it controls the overall functioning of a cell. That is because, suppose so how protein is made. So when, suppose I am drawing a ribosome over here, if I draw a ribosome over here, so ribosome, in order to make a certain you know mm, a certain enzyme, what ribosome, what the DNA does is, it will send a typical formula, a one stranded DNA, right? It will send a one stranded DNA to this ribosome, and it will make a particular enzyme. For example, suppose DNA wants to make an enzyme A, or 1, right? So maybe it will clip this portion, okay, and send it to ribosome. So ribosome can only read a single stranded DNA, right? It can read only a single stranded DNA. So or maybe for example, DNA wants to make an enzyme too, so maybe it will clip this portion, and it will give it to a ribosome, right? So now ribosome will take that formula. For example, suppose it has taken A, G, T, 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 C, okay, and from here, by reading that formula, ribosome will make a particular enzyme, right? So this one stranded DNA is known as RNA, right? This one stranded DNA, we will call it as RNA, ribonucleic acid. Why ribonucleic? Because the oxygen atom is present over here, oxygen molecule is present over here, right? So so what a ribosome does is, ribosome will take one stranded RNA from here. uh, what happens is, ribosome will make a particular enzyme. Ribosome will read that formula, and it will make an enzyme. So this from DNA is making RNA, right? So this process is known as transcription. Sorry. And from RNA to ribosome, and finally forming an enzyme, this is known as translation, right? So I hope that was clear. So this is how DNA controls everything, and that is why it is called the brain of the cell, right? So now let's move to the difference between a prokaryotic and a eukaryotic cell. Okay, so now let's understand the difference between a prokaryotic and a eukaryotic cell. So we'll make a small table, and so here we'll take prokaryotic. Here we'll take eukaryotic. What does pro mean? So pro means before nucleus, where there a true nucleus is not present, whereas in eukaryotic cell a true nucleus is present. For example, in prokaryotic, in eukaryotic cell we saw earlier here, we can see we have a proper, a membrane, right? It is inside a proper nucleus. But in an eukaryotic cell, the genetic material, they float randomly within the cytoplasm. I mean, inside the cell, inside the cell membrane in the cytoplasm, right? So the first difference what we can notice is, so the first difference, as I said, just a second, first difference, as I said, here we'll have no defined nucleus. Here we have a defined nucleus. Here in eukaryotic cell, sorry, in prokaryotic cell, the genetic material floats randomly. Here there is, in prokaryotic, there is no compartmentalization, right? The cell, or the cell organelle, or the others as well, they float randomly, as I said. For example, here we can see everybody is, I mean, every organelle is inside a proper membrane. There is strict separation. But in a prokaryotic cell, those compartmentalization are not there, right? So then, prokaryotic cells are generally, small in structure, whereas eukaryotic cells are generally larger. Here, one difference is they are generally unicellular. They can be multicellular. For example, humans are eukaryotic, basically, right? So so the type of chromosomes. So what are chromosomes? We didn't discuss it beforehand. So chromosomes are basically, we'll discuss it later as well, but just a small, glimpse that, beforehand I said, the genetic materials are present inside nucleus. So inside nucleus there is chromosomes, and inside chromosomes there are genetic materials, for example DNA and RNA. We'll go into detail in chromosome in, I mean, either in today's video or in the next video. So up till then, let's see. So the chromosomes in prokaryotic, they are generally, one single loop. Okay, there is generally one single loop of DNA and plastids, whereas in eukaryotic cell there are generally more than one, right? What is the case of ribosome? So as I said earlier as well, ribosome is a non-membrane bound, right? So of course it will be present in both prokaryotic and eukaryotic cell, right? And prokaryotic cells are generally, they are less complex, their structure is less complex, whereas eukaryotes are have more complex structures. So thus their replication or their growth, prokaryotic cells growth are, very fast. It can replicate quite easily, it can multiply quite easily, whereas since these eukaryotic cells are much complex, their growth is, slower than that of the prokaryotes, right? So yeah, these were some of the differences. Let's see some examples. So pro bacteria, okay, bacteria and archaea, they are both examples of prokaryotes. We have other examples as well, but let's settle with this only. And in eukaryotes we have animals, all sorts of animals. We have plants, we have fungi, we have protist cells, and etc. We have others as well. So these are some examples, and now we'll go. We'll study in depth about some organelles. For example, we'll start with nucleus, cytoplasm, and ribosome. Okay, so nucleus. So nucleus is a dense membrane bound structure, right? Dense membrane bound. And as I said, in prokaryotes, that membrane is not present. The DNA material floats randomly, and inside nucleus we have chromosomes, and inside chromosomes there is DNA and RNA, right? So next up, let's take cytoplasm. Inside, I mean, it is a semi-fluid. It is a semi-fluid structure. Then present, of course, both in pro and eukaryotes. We have seen in the first slide as well that in order to call something a cell, it should have cell membrane, then it should have cytoplasm, right? So of course, both prokaryotic cell and eukaryotic cell will have cytoplasm. Then the main function is chemical reaction. Every activity happens in that fluid matrix only, inside the cytoplasm, right? Now let's come to ribosome. As I said, it is a non-membrane bound structure present both in prokaryotes and eukaryotes. Okay, so yeah, so now we have cell membrane. Okay, so we have cell membrane. So cell membrane is, let me write it first, it is selectively permeable. Okay, selectively permeable means it's not that it restricts everything, as well as it does not allow everything as well. Okay, things that are necessary, those things will be allowed into that particular cell, so that is a function of cell membrane, right? So they are made up of lipids plus proteins. They are quasi fluid in nature, right? They are quasi fluid in nature. So if we draw a cell membrane, so it will look something like this. Right. So these are lipids, and these are phosphates. So phosphate is generally written like this, PO4 3 minus. Okay, and so this entire thing, okay, this both, or sorry, if we take the single one, so then we'll call it as phospholipid. And if we take both of them, we'll call it phospholipid bilayer. Where should I write it? Okay, phospholipid bilayer. And what are these small? And this is peripheral protein. Sorry. And this one is integral protein. Right. So this one is upper one is peripheral protein, and this one is integral protein. This integral protein is, it is properly binded, so it is very difficult to be removed, right? So the main purpose of this cell wall is there are three types of transportation which occurs, right? So it is active, passive, and there is osmosis. So in active, not everything can enter. things can enter, but a certain amount of energy will be required, right? In passive, so there is no energy will be required in passive transportation. Things can enter easily, but with osmosis is passive transportation only, but with the help of water. The medium should be water, right? So now we'll come to cell wall. We are done with cell membrane. We'll be doing cell wall. Uh, so what is the function of cell wall? Cell wall is cell to cell interaction, and in cell wall, cell wall are not present in animals, right? So it is present in plants, fungi, algae, prokaryotes. So yeah, and if you see the structure of a cell wall, it is generally like this. There are small passages. It is a random structure. It is not very precise. That happens. So basically, this is how the cell wall looks like. This covering is rigid covering, that is it is non-living in nature. So yeah, so the main function of cell wall is cell to cell interaction only. So there are various compositions. For example, for algae, the cell wall will be made of cellulose, then galactans and mannans. For plants we have cellulose. This is for plants. We have cellulose, then we have hemicellulose, we have and proteins. In case of prokaryotes, peptidoglycan is present, right? These are the composition of several cell walls of different there was a previous mains question as well regarding this composition. So these things become very important from that perspective. So now let's come to mitochondria. Right. So in mitochondria, what happens, as we have said earlier as well, here aerobic respiration takes place, where we have seen C6H12O6 plus oxygen giving rise to energy and carbon dioxide and everything else. So yeah, the main function of mitochondria is aerobic respiration, and the energy is produced in the form of ATP, right? Adenosine triphosphate. So another interesting thing is the mitochondria has its own singular circular DNA. So up till now we have read that all the genetic materials are only present inside a nucleus, but this mitochondria and another organelle as well, we'll read it further. They have their own DNA as well. So what is aerobic respiration? Aerobic respiration, we saw that when respiration is happening in the presence of oxygen, and then when energy is produced, then it is called aerobic respiration. But when respiration is without the presence of oxygen, then it is anaerobic respiration. For example, when we are running, so we feel certain cramps in our body. That is because body is producing energy without the presence of oxygen. That is the reason we tend to inhale so fast, right? And that is the reason we have cramps. So cramps are due to the formation of lactic acid when body's respiration is anaerobic. Energy is produced with lactic acid is also produced, and that is the thing that helps that produces that those cramps in our body, or in our legs for that matter. So generally what people say is to pour lukewarm water, or to take a hot bags. What does that help? So what it does is it dilates the blood vessels so that more oxygen can enter and it can mitigate that lactic acid effect, right? So yeah, so that is anaerobic respiration. So in mitochondria, aerobic respiration happens, right? And there is another information, that is mitochondria is double membrane bound, right? It is double membrane bound, and it has, as I said as well, it has own single cellular DNA. So yeah. In mitochondria, we just saw that the mitochondria has its own, single circular DNA. So, what other organelle is there where this genetic material is present? As of now, we have seen in mitochondria and in nucleus, genetic material is present. So another place where it is present is in chloroplast, right? So in chloroplast, in mitochondria, and in nucleus, their genetic material is present. So how come here in these two organelle we see the genetic material? So there is a theory known as endosymbiotic theory, or endosymbiosis, where what happened was thousands of years ago a bacteria started to live inside a cell, right? So in due course of time, that cell became an organelle, right? And thus the ribosome and chloroplast, they started they got their own genetic material. So initially it was believed that let me write this down first. Endosymbiosis, or endosymbiotic theory. So initially it was believed that nitrogen fixation happens only in bacteria and archaea. Okay, but there was a recent study which suggested that which showed that an algae can also do nitrogen fixation. In an algae, there was an organelle was found named nitroplast, and through which nitrogen fixation could be done. Yeah, it can do its own nitrogen fixation. So yeah, so that was how this chloroplast and mitochondria started having their own genetic material. Okay, now we have a small section left, that is genetic material. uh, we studied we studied it previously as well in depth. So now I'll just tell you in short. Functions of a genetic material. So the first thing which it will do is replicate. It will replicate itself, right? So first it will replicate, replication, right? Second, it is a storehouse of information. It is chemically and structurally stable. Okay, chemically and structurally stable, So that is the reason with with DNA testing you cannot tell the age of a person, because they are chemical and structurally stable, right? With age, genetics doesn't change, right? So the main function of this is to store information, right? And so the changes which occurs, that is occasional, that is very slow changes, that is called mutation, right? So yeah, so now previously I've said that inside the nucleus there is chromosome, inside then in chromosome there is genetic material. So what happens is, so the what is the length of an entire DNA? It is approximately equal to 2.2 meter, right? Its length will be approximately 2.2 meters, right? So what happens is, so there are thousands and billions of DNAs that will be present in our body. So how will that fit in? So they get coiled. So the structure in which they get coiled is a protein known as histone protein. Okay, so they coil themselves around that histone protein. So that coil structure is called chromatin. Okay, so from DNA coiling we are getting chromatin, and when it coils further, so densely coiled chromatin is chromosomes, right? So yeah, they look something like this, right? They look something like this. So yeah. So anyways, that was it for today, guys. Next class we'll see about human reproduction, and there we'll study more about chromosomes. We'll see various things: haploid, diploid. There is mitosis, meiosis. How many? So anyways, that was it for today, guys. Thank you. Goodbye.